Chapter 23 covers protists. We'll examine their eukaryotic origins, their general characteristics, how they're grouped together, and the ecology of those individual groups. The body forms of protists are highly variable. Some are unicellular with few physical characteristics that make them appear more than just a little solitary sphere. Some protists are ciliates and covered in long cilia that can drive both their own movement and the movement of their food into feeding grooves. The largest protists are hundreds of feet tall, or you could say long, and exist in massive underwater kelp forests. Just as the rainforests on land create their own little worlds, these kelp forests do the exact same thing in expanses on the ocean floor. This list obtains the defining characteristics of the eukaryotic organisms that exist in kingdom Eukarya, in which we find domain Protista. Modern members of the Protista domain have these characteristics now or have their hallmarks or vestiges of these characteristics from their ancestors. Endosymbiosis has been discussed in this book previously. It's the idea that a composite host cell organelle engulfed some alpha proteobacteria that then took up residence inside of it. Their symbiotic relationships can actually be tied to the metabolic diversity we see today in Protista. The organelles that were once independent individuals have shared their taste for particular resources and have given eukaryotes some unique abilities. Having been outcompeted in a lot of instances, though, we do find that most eukaryotes tend to be aerobic. We'll meet a few individuals in Protista, though, that are definitely breaking this mold. Eukaryotic cells may contain anywhere from 1 to several thousand mitochondria, and each mitochondria can be anywhere from 1 to 10 micrometers in length. Each is considered a single mitochondria, but they can fuse together to form one larger mitochondria. The interesting thing about their division, though, is that cells can't actually make their own mitochondria. Mitochondria can only form from other mitochondria. They contain their own separate DNA. They're independent from their host in their replication, which is a throwback to the fact that they were brought into the cell via endosymbiosis. They have, however, existed in cells for so long that they can't actually survive outside of their hosts anymore and are considered just a normal organelle, be it an interesting one. Small packets of pigment known as plasmids have been brought into cells via endosymbiosis too. If the pigments are capable of undergoing photosynthesis, they're referred to as chloroplasts. They seem to have evolved from a cyanobacteria-like ancestor, and as mitochondria do, they often carry their own DNA. They have a single circular chromosome and even ribosomes of their own that help them undergo the process of photosynthesis. Not all plasmids in eukaryotes are derived directly from primary endosymbiosis. Some of the major groups of algae actually became photosynthetic via secondary endosymbiosis. That is, they took in green algae or red algae as endosymbionts, so they ate someone who had eaten someone who was carrying a chloroplast. Numerous microscopic and genetic studies have actually supported this conclusion. Secondary plasmids are surrounded by 3 or more membranes, and some secondary plasmids have even clearer remnants of the nucleus of the original endosymbiotic algae. Others have not actually kept any of the remnants. These are cases where higher order endosymbiotic events are the best explanation for plasmids in some eukaryotes. The first eukaryote may have originated from an ancestral prokaryote that had undergone membrane proliferation, compartmentalization of cellular functions, and establishment of endosymbiotic relationships with an aerobic prokaryote, and in some cases a photosynthetic prokaryote, to form mitochondria or chloroplasts respectively. So what that means is that there were infoldings of the plasma membrane that gave rise to different compartments. Within those individual compartments, different cellular functions may have evolved. In the case of lysosomes, helpful enzymes would have been sequestered into protected spaces. In the endoplasmic reticulum, RNA would have been given the opportunity to create proteins, and those proteins would have been given a protected space in which to fold. Secondary endosymbiotic events would have brought in batteries, essentially, in the form of mitochondria or chloroplasts, and modern heterotrophic eukaryotes would have been the result. This might seem like a lot of ifs and thens, but you have to remember these processes were undergoing trial and error for billions of years with absolutely nothing to get in their way. The hypothesized process of endosymbiotic events leading to the evolution of chlorokineophytes is shown. In a primary endosymbiotic event, a heterotrophic eukaryote consumed a cyanobacteria. In a secondary endosymbiotic event, the cell resulting from that primary endosymbiosis was consumed by a second cell. The resulting organelle became a plastid that's seen in a modern species. The characteristics of protists can be fun to study because they're so diverse. The cell structure is highly variable and can be beautifully elaborate. They can be tiny or huge, soft or structured, silica-wrapped or even protein-stripped. When it comes to metabolism, some are aerobic, others are anaerobic. Some protists move with cilia, while others pull themselves along with pseudopodia, and others still with whip-like flagella. Their life cycles may be sexual or asexual depending on the species, and some life cycles can be long, elaborate, drawn-out processes. As for habitats, they can be found anywhere and everywhere on our planet, maybe even on some others. Let's focus on the metabolism for a minute. Now, some protists are simply phototrophic and undergo photosynthesis. Others are saprobes and absorb material from decaying matter. Here you can see the process of phagocytosis, which is when heterotrophic organisms take in food and digest it internally using some hydrolytic enzymes that exist inside of their lysosomes. Some protists can function as mixotropes, so they obtain their nutrients by a mix of photo and autotrophic or heterotrophic routes depending on whether there are sunlight or different organic nutrients available. These are truly masters of their trade. Here's another image to help you get some of those terms straightened out. The paramecium in A is covered in cilia, or short hair-like projections that can be in unison or in groups. The amoeba in B has pseudopodia, or false feet, and they can actually help it crawl around or pull larger items towards its central body. Finally, the euglena in C has a long whip-like flagella that helps it to propel itself through its aqueous environment. The supergroups of eukaryotic organisms are listed in this phylogenetic tree, like the bacteria and archaea were for the previous chapter. Here we see three groups that we'll discuss in detail. They're Excavata, Chromia alveolata, and Rhizaria. It would be wise to write down these names in order as we're going to take a close-up look at all of the different phyla, clades, and some more specific species within these supergroups. Let's take a look at the other half of the list first, and then we'll start discussing the Excavata. Three more eukaryotic supergroups can be seen here. They are Archaea plastida and Amoebozoa, and then the Opisthocontas. The first supergroup of Protista discussed are the Excavata, which are largely asymmetrical single-cell organisms that have a feeding group along their body structure. Some are predators, others are photosynthetic, and some are even parasites. There are three major kingdoms under Excavata. The diplomons are seen here. In this example, you see Giardia lambia. This is a waterborne parasite that causes severe gastrointestinal distress. The second kingdom that was listed under the Excavata supergroups are the parabalsalids. These protists function anaerobically and are called hydrogenosomes after an organelle that they have. The organelle produces hydrogen gas as a byproduct from a special aerobic respiration process. They move with flagella and have kind of a signature membrane rippling. One of them is known to cause the STI trichinomonas. A third excavata are the euglenozoans. Euglenozoans include parasites, heterotrophs, autotrophs, and even mixotrophs. They range in size from 10 to 500 micrometers. Euglenoids move through their aquatic environment using two long flagella that guide them towards light sources that are sensed by an ocular organ known as an eye spot. A famous subgroup of the euglenozoids are called kinetoplastids. They're named after their kinetoplast, which is a DNA mass that's carried in a single oversized mitochondria. T. brucei is a specific species that's well studied because it causes African sleeping sickness, and it's considered a euglenozoa. Sleeping sickness is a major issue in certain parts of the world, and its life cycle, which is seen here, is of particular interest because it's actually easier to stop the transmission of the disease by stopping the bites from the tsetse flies than it will be to cure the disease itself. So in this instance, it's important that entomologists work with epidemiologists and doctors in the field. The second supergroup on our list was the chromoalveolata. Chromoalveolata are known for secondary endosymbiosis. It's thought that they formed from an algae and some other biont or living form, but we're not entirely certain. The defining characteristic for this group is the presence of chlorophyll C. One group within chromoalveolata are just the alveolates, and they have a membrane-enclosed sac underneath their cell membrane. There are 3 common phyla that are all considered alveolates. The first are the dinoflagellates. They have 2 interlocking plates of cellulose and 2 flagella that give them a spinning motion as they move through the water. They're also quite famous for their bioluminescence, which we'll see in just a moment. Another common phyla are the apicomplexans. These have microtubules, fibrin, and vacuoles that are all asymmetrically distributed, so it gives them a funny shape. All species under this phyla are parasitic. Finally, we have the ciliates. Ciliates range from 10 to 3,000 micrometers in length, and they're covered in rows or tufts or little tiny spirals of cilia. They can withstand some pretty intense environments because they're wrapped in a pellicle, which gives them a hard outer covering on the outsides of their bodies. Here's an artist rendition of the dinoflagellates. They have incredible diversity in their shape, but are known for their 2 flagella that are situated on different planes, so they're perpendicular to one another. Their movement causes the dinoflagellates to spin through the water like a kid's toy top across the floor. Here's their characteristic bioluminescence as seen from the coast. This property is thought to be tied to changes in the pH, but it's also useful to scare off predators. The second phylum was the apicomplexans. They have a specialized organelle to help them infiltrate host cells, and all of them are parasitic. They're also all unicellular and spore-forming. An apicomplexan called Plasmodium causes malaria. You can see its complex life cycle here. Like sleeping sickness mentioned previously, it's actually easier to stop the transmission of the disease than to cure it. So aid workers are currently focused on cutting out the middleman of the infection cycle, that's the mosquitoes, in order to combat the transmission of malaria. Final alveolate is the phylum of the ciliates. Paramecium are ciliates that are found in almost every water source. They have a primitive mouth into which they can sweep their food with their cilia, and an anal pore to eject waste, and a contractile vacuole through which they can remove excess water. Their body plan is busy, as is their sexual reproductive process. There are 2 different mating types seen here. The body plan of Paramecium are busy, and as is their sexual reproductive process. There are 2 different mating types that can come together and form a cytoplasmic bridge. They each undergo meiosis, making 4 haploid micronuclei, but only the best are kept. The remaining nuclei divide via mitosis and then swap one of their newly formed nuclei with their neighbor. They then fuse together and undergo more rounds of mitosis, reproducing themselves 8 times. Each individual then loses their original macronuclei and divide twice to form 4 new cells. It's quite the process. Underneath the supergroup chromoalveolata was a listing for the stromatophiles. These individuals are photosynthetic marine algae or heterotrophic protists. Many of them also have hairy flagella, and there are 4 phylum that we're going to look at. The diatoms are unicellular photosynthetic organisms that have silicon dioxide in the matrix of their organic particles, making them quite firm to the touch. Golden algae are largely unicellular too, and they contain a lot of carotenoids, which gives them their signature yellowy gold color. They exist in both freshwater and marine environments. Brown algae are primarily marine and tend to be multicellular. Some of them look a little bit like terrestrial plants, although there are a lot of differences. Oomycetes are water molds that are fungus-like in their morphology, but they have cellulose-based cell walls, just like regular land plants do. This picture shows you a collection of diatoms. The silicon-encased shell appears to glow bright blue in this image. These guys live in saltwater environments. Here you can see an example of brown algae off to the left, and then its life cycle, which is actually very similar to that of many, many other plants. A single sporophyte individual who is 2N, creates spores that are capable of growing into male and female gametophytes. The gametophytes then grow normally and create egg and sperm, which fertilize each other to form a new sporophyte individual. This idea of living as both haploid and diploid separate creatures, it's considered the alternation of generations. This picture shows you the great decayer of the world, the oomycete. The mold is engulfing and breaking down an old insect body. It can return resources trapped in dead bodies and bring them back to the world of the living. Moving on from the chromo-ovulata group, we enter into the Rhizaria supergroup. Rhizaria includes amoebas, most of which have thread-like or needle-like pseudopodia. Pseudopodia function to trap and engulf food particles and to direct the movement in Rhizarian protists. The protist then transport its cytoplasm into the pseudopod that's reached out, therefore moving the entire cell. This type of motion is called cytoplasmic streaming, and it's used by several diverse groups of protists as a means of locomotion or as a method to distribute nutrients and oxygen throughout the entire cell body. Forams, a type of Rhizaria, are unicellular heterotrophic protists, and they range from approximately 20 micrometers to several centimeters in length. Sometimes they can even form structures that look like teeny tiny little snails. They have porous shells called tests that are built from various organic materials and typically hardened with calcium carbonate. The tests can actually house photosynthetic algae, which the forams then harvest for nutrition. Foram pseudopodia can extend through their little pores and allow the forams to move or feed or gather additional building materials. Typically, these guys are associated with sand or other particles in marine or freshwater habitats. They're really useful indicators of pollution and changes in global weather patterns, as they should exist everywhere, but if the health of the water is affected, you see it in the forams first. A second type of Rhizaria, the radiolarians, exhibit really intricate exteriors of glassy cilia with radial or bilateral symmetry. Needle-like pseudopodia supported by microtubules radiate outward from the cell bodies of these protists, and they function to catch food particles. The shells of dead radiolarians sink to the ocean floor, where they can accumulate up to 100 meters thick in certain depths. Preserved sedimented radiolarians are really common to find in the fossil record, so they're evolutionarily quite old. Supergroup 4 on our list began with the archaeoplastids. They include red algae, chlorophytes, charophytes, and land plants. It's from a common ancestor of these particular protists that land plants most likely evolved. Molecular evidence supports that all archaeoplastids are descendants of an original endosymbiotic relationship between a heterotrophic protist and a type of cyanobacteria. Red and green algae include unicellular, multicellular, and even colonial forms. Volvox aureus is a green algae in the supergroup Archaeoplastida. This species exists as a colony and is thought to maybe be one of the first semi-unicellular organisms. It contains cells that are immersed in a gel-like matrix and intertwined with one another via hair-like cytoplasmic extensions. So while each cell could technically be argued to be a separate individual, they depend on one another to survive. Chlorophytes, as I mentioned, inhabit freshwater and damp soil and are a really common component of plankton. They're a subset of green algae. This example is an interesting one. It's unique because it's actually a single cell. It just looks like it's organized into leaves. There's no vascular system supporting water through that plant. So even though it looks advanced, it's really not. That's one huge branched cell. The sixth listed supergroup are the amoebozoans. The amoebozoans characteristically exhibit pseudopodia that extend like tubed or flat lobes, rather than the hair-like pseudopodia of Rhizarian amoeba. The amoebozoa include several groups of unicellular amoeba-like organisms that can be free-living or, of course, parasites. The life cycle of a Plasmodium slime mold, a very popular amoebozoan, is shown. The brightly colored Plasmodium on the bottom is just a big multinucleate mass. So you can see the original sporophyte that has a tall stalk and a sporangium that has created spores through the process of meiosis. Germination gives rise to cells that can actually switch back and forth between being amoebas and flagellated zoospores. Fertilization occurs when two spores find one another and results in a 2N zygote. That 2N zygote grows via mitosis, but never actually goes through that cytokinesis stage to develop more than one cell. So you just end up with a multinucleate mass, and that brings us back to the plasmodium that we saw in our original picture. Slime mold may exist as solitary or aggregated amoebas, and these are pretty interesting looking. There's one that's actually called a dog vomit slime mold, so they're all not that great. The life cycle of a cellular slime mold is similar to the life cycle we just saw. You have an aggregate stalk with a fruiting body that releases 1N spores, and germination of those spores gives rise to amoeboid cells. They can grow via mitosis or meiosis to create either a 2N individual or a 1N individual. And mitosis continues on without cytokinesis, so you end up with this big mass of aggregated cells that are all multinucleates. Kind of gross, kind of cool, not something you see very often, especially not here in the desert. The final group is the Ophistocontas. They're animal-like cyanoflagellates that are evolutionarily related to all sponges and animals. They exist in unicellular or colonial forms. There are about 244 described species. They have singular apical flagella and are surrounded by a contractile collar composed of microvilli. That's something that we're going to see in sponges when we get to animal diversity. Now that we know a lot about the diversity of protists, let's look at why they're important to the environment. Protists are actually vital to the environments in which they're found. They're often a major food source and the base of food chains wherever they exist. Some fix inorganic carbon into useful organic carbon that gives the fragile life forms on which they live a boost of energy. A great example of this would be corals. They feed corals carbon, and corals in the meantime protect a lot of the protists from being eaten. This is considered a symbiotic relationship because the two creatures help each other. The protists themselves and their products of photosynthesis are essential, directly or indirectly, to the survival of organisms ranging from bacteria all the way up to mammals. As primary producers, protists feed very large portions of our world's aquatic species. In fact, approximately 1/4 of the world's photosynthesis is conducted by protists, particularly dinoflagellates, diatoms, and multicellular algae. The fact that so much of our energy is coming from these protists living in the oceans is the reason that we have to be extraordinarily concerned about any potential pollution or rapid shifts in temperature. Protists, of course, are not always helpful in a given environment, and some are very famous pathogens. They cause illnesses like malaria, African sleeping sickness, and waterborne gastroenteritis in humans. Here you can see the causative agent of malaria, which is growing inside and around these red blood cells in the affected patient. This is African sleeping sickness, which we've talked about before. The mature parasites can be found in the blood of affected individuals. Here's another example of a reason that protists can be incredibly dangerous. You can see a downy and a powdery mildew found on these grape leaves, and it's been caused by an infection from something called P. viticola. Another common and upsetting infection is caused by P. infestans, and it was the causative agent of the potato blight, which led to the death of thousands and thousands of Irish citizens. Scientists are actively working to control the infections caused by various protists to attempt to protect our food resources around the world. With that note ends chapter 23 on Protista. Go through, read the chapter, take notes in your own words, do practice problems, do some more practice problems, and then take a crack at your homework.